Thanks everyone so much for joining us for the Applied Research and Education Seminar. I think all of us know the goal of the seminar is to bring together data scientists to discuss interdisciplinary research, and it's run by Kenney, Ontario, and all the partner institutions. So I'm Jessie Gronville. I'm an assistant professor at U of T, and I'll be hosting the talk today. Before we get started, I just wanted to say the land acknowledgement. We wish to acknowledge this land on which the University of Toronto operates for thousands of years. It has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. Okay, so with that, I'm excited to introduce Dr. Anna Kenney. She's an assistant professor. She just started at the University of California at Irvine. She works at the interface of statistics, interpretable machine learning, and large-scale optimization to advance biomedical research, while also participating extensively in collaborative interdisciplinary research. Before joining UC Irvine, she was a postdoctoral scholar at UC Berkeley Statistics with Dr. Bin Yu, and she completed her PhD at Penn State Statistics and Operations Research, where she was a biomedical big data to knowledge training fellow, that's a mouthful, and Alfred Sloan PhD scholar. Today, she'll be talking about leveraging problem structure to improve future recommendations in biomedical research. And her preference is you may ask questions during the talk, so feel free to raise your hand, and I'll also be monitoring the Zoom.
Thank you.
Please take it away. Thank you.
Thank you all for coming and for the invitation. I was very happy to get to come in person and visit Toronto again, even if it's November and I'm coming from Irvine. So as mentioned, let's see. So this is going to be work that was conducted over the course of my postdoc, mainly with people in Bin Yu's group and some collaborators. And so given the kind of flavor of the ARES seminar, I didn't want to just give like stat methods talk, but I also wanted to show sort of the domain problem that led to some recent advances that we pursued in random forest feature affordances, right? I think it's very key to give credit to my collaborators, as well as where a lot of cool stat methods really come from, and that is from very problem-centric data science work. And so I was fortunate to be a part of several collaborative projects. The one I'll be focusing on here are the episodic drivers of cardiovascular disease. I want to shout out all my collaborators. And so the challenges that I've seen and that I think a lot of us here have seen and speaking with the students are that when you work on these domain-centric problems with collaborators, you're going to see a lot of combinations of challenges you're maybe not used to, and that a lot of existing methods will simply break apart when you try to get started, right? Just things start failing very quickly. And so, for instance, here we're going to see issues with dimensionality. We've dealt with data contamination. So if you work on very noisy, publicly available data sets, there could be measurement error involved, right? Correlated features, different types of responses, and especially in the biomedical setting, data privacy is a concern. And so, well, I'm going to focus here on random forest. I think you should sort of be incentivized to not maybe lock yourself down to a specific type of method, because I think depending on the problem, it's going to call for different areas of statistics and machine learning, hence how I kind of fell into a few of these pots here, right? But regardless of the method you kind of focus on or the coolest new predictive model you get to work on in your subject area, let's say statistical genomics, there's oftentimes this question when speaking with your collaborators, especially where they're asking you, okay, but what do we do next, right? You have this great new predictive model. I don't know what to do with that. I know that you took genetic data and you put it in, but now what do we see? And so oftentimes the next step might come in the form of clinical trials or actual experimentation. So in this case, I was really fortunate and also anxious about being part of a project that at the end of the day, they were actually going to take our recommendations and perform gene silencing experiments. So if you think of like CRISPR, where you're actually editing certain genes, similar, what you're doing is you're suppressing certain genes and seeing the effect on a given type of cell, for instance. And so these are very costly in nature. And so it's not like you have many chances to keep going back and forth, right? And say, okay, sorry, we messed up. We're going to try a new approach. Here's the next guy. I hope this one works. So you have to really carefully not only try to create these stable models and analyses, but how do you sort of simmer that down to a smaller set of things for them to look through? Because you only have so much funding basically to do these types of tests. So it was the first time for me, at least, I kind of encountered very practical issues in terms of like, how can we actually test what you're finding and validate them, right? And so I have to give credit to my postdoc advisor, Professor Bin Yu, who, if you guys haven't read it, there's a nice paper on veridical or truthful data science that goes into a lot of the judgment calls we might see as statisticians approaching analyses, right? And so what are ways we can kind of try and prevent, pursuing one particular pipeline because it seems to work well? How do you balance this out well so that you have more stable results downstream? Okay, so on this talk, I'm going to start with this epistatic drivers project and then get into how that incentivizes this new unifying framework for random forest feature importance. And so the MDI, both papers are now up on Biomed Archive and Archive in case you're interested in seeing more details. And so just to start, because especially for people who aren't in larger collaborations, it's fun to show just how much work goes into it and everyone involved. So this is from multiple institutions funded through the Chan Zuckerberg BioHub Fund. And it started off with our main PIs, Bin Yu and Ewan Ashley in Stanford, as well as James Priest, who came in later. And then it started off with just two students, Tiffany Tang and Chandra Wing at Berkeley and Stanford, before myself and others joined at different points in the pipeline. And everyone was important to getting this to where it is today, where we actually saw validation and experiments and had different roles to play throughout the pipeline, as I'll discuss. But so the overall idea here was to find genetic drivers contributing to cardiovascular disease. We played to our strengths. Professor Ewan Ashley and his lab are fantastic experts in cardiology with a focus on things like HCM, which is a known sort of genetically, a known disease in the heart that we have pretty clear genetic drivers for. And so CVD is a leading cause of death globally and in the US, but there are also other environmental or risk factors that might contribute to it. Poor diet, physical inactivity, tobacco use. During my job talk, I said Tabasco use, but it went over very well and I got the job. So I like to mention that now. So it's okay to make funny mistakes during these talks. But there's certainly a gap in the literature, as those of you who are now in the stat genomics class have probably seen, where we do genome-wide association studies. I as well have constructed things like polygenic risk scores. But based off current heritability estimates of a given disease, there tends to be this heritability gap as it's listed in literature. And so what that means is, and this was from a 2022 paper, that heritability might be estimated to be, say, between 40 and 60%, but existing models based off, say, GWAS might only cap out at around 15%. So there's sort of this question of like, all right, well, where is this coming from? What exactly are we missing in our analysis? Is it just a data issue? Are we not considering everything we should? Is it a modeling issue? And so some people think that, well, if you focus more on interactions, you might help close this gap versus just considering marginal effects on a model and actually see how different genes might interact. Others might say, oh, you need to look more into these rare variants or things that are in coding regions. And so here we focused on, okay, what type of power can we gain by looking at epistatic interactions? Does that validate in an actual experimental setting? But there's actually a lot of debate just on this heritability issue that I won't dive into here. There's even arguments over whether our current heritability estimates are overdoing it. But I'm happy to talk about that with other people who are interested. But here we're going to focus on the epistasis component. And so the goal for this project was to try and develop a gene interaction recommendation system and then follow through with wet lab experimentation. Okay. So the data that we had is publicly available, and I think there's some really cool initiatives happening here to where maybe more of you can gain access as well that I heard about. That's UK Biobank. And so here our phenotype of interest involved 30,000 patients from a white British unrelated population with cardiac MRIs. So we're actually looking at these short little videos of the heart beating, and what we're taking from it using what I'm just going to call deep learning techniques, meaning so you go and trace the heart, you find the walls, and you measure the thickness of that wall over the course of the video, okay? And so if you have a thicker heart wall, this can contribute to other risk of like heart attack and disease, right? And so that's what we're pulling out from these videos. And this was mainly led by Weston Hughes to do that extraction. And the genetic features that we have are 15 million SNPs, which of course come in pretty high correlation blocks. For those unfamiliar, so we're having a very wide matrix, so only around 30,000 observations with 15 million features, right? And each feature takes on values of 0, 1, or 2 for like the number of alleles there. So like most things, you can't just apply and shove that data set into your, toolbox of models. And so the challenge is just right off the bat, looking at this problem, dimensionality, can we trust the data that we're getting? We're using a model just to extract the phenotype in the first place. So there's also potential model noise there. Weaker signals are expected with these types of complex diseases. And the response itself, you could even model in a smarter way with like, can you look at the way the thickness changes over the course of the video, for instance? So even at this step, there's already judgment calls being made, right? We actually binarized this measure of thickness so that we can look at comparing extremes so that we're denoising the problem a bit. But there are many ways just to go different routes from this step itself. Okay, so I do want to get to the stat and method component, but I want to run down just a quick idea of how this pipeline works to tie it back into random forests. So with our pipeline, many of you may have guessed that there's no way I'm going to put a 15 million feature matrix into a random forest model, right? It's not going to work. And so we still use GWAS. And we use two different approaches that were pretty common, Bolt LMM and the default in Plink, but I think they're both mixed model approaches anyways. And so what we did is we tried to insert some stability by looking at the union across the top hits for both. And so that led us to around 1,400 SNPs. So as you can see from the GWAS plot itself, you're not getting a lot of guys that are going to cross over your standard threshold. And the ones that do, of course, are centered around Titin, which is a very well-established gene connected to cardiovascular phenotypes in general. So this is kind of a sanity check. We also know that, of course, we're going to get a lot of hits on Titin. It's not the most interesting, but we're more curious about what else we can pull up. So by also going down to 1,000, we're getting more that did not pass the GWAS threshold, but we're putting all that downstream into another model to then filter. Okay, and so the approach we did to try and tease out interactions, because if you just try to consider every possible interaction of even 1,400 features, right, that's a lot to work off of. And so we use iterative random forests. And so the idea behind RRF, which is shown to be effective in this type of setting, is that you're, again, refitting random forests across multiple iterations, but instead of just randomly from a uniformly sampling from your features, that's the random part of a random forest, you reweight each feature based off of the feature importances given on the previous iteration. So you run your random forest, you get an idea of how important different features are, you then use those in the probability to try and resample with weights given, higher weights given to more important features, okay? And you do this repeatedly until it passes some stability criterion, basically. And then you go ahead and take a look. I put an alert here because turns out your default feature importance measures in random forests are not great for this setting. And we'll see why in a bit. But putting a pin on that, the interactions found are basically looking across all of the trees that we see and saying, oh, okay, if I split on X1, I always tend to split on X2 next, right? So you're trying to find these little chains of features that commonly are co-occurring together. So the idea is if they're always like seem to be splitting together in some way, maybe there's some relationship there, some interaction present. Now, I'm not talking about any theory establishing consistency of finding these interactions. This was strictly empirically sort of based. There was some light theorems in the original paper, but for the most part, it's empirical work, really using intuition of like, I'm seeing these split together. I think that might mean something. Let's see how it works. And is it predictive? Okay. All right, so we use the results from IRF to essentially get some nice ranking of not only the features, but also interactions between features. And so what we did that's a little bit different is we did local importances, meaning we looked at for a given patient, what are sort of your top hits, right? So you might have a different set of SNPs compared to others. And so that way you can see just how stable this is across patients, or do we see kind of clustering among patients where certain people had these features as most important and other people's had different features more important. Now, the UK Biobank, after all these filtering, is fairly homogeneous, so we didn't see big differences, but this is just to say it might be helpful for you to think about local things as well, because you might have hidden subgroups in your data, right, that maybe you didn't account for. All right, so our result at the end are 3 genes and 2 stable interactions. And that's not because they were the only things that came out. This is from a list of things, okay? And so the reason we have such a small number is because at the end of the day, we're talking about doing gene silencing experiments. So we actually could only test like 5 things due to budget issues, right? So essentially it was, these are seemingly the most stable, the most predictive. We're hoping for the best. And now we're going to go ahead and test it. All right. Yes.
For the 1400 SNPs,
Yeah.
GWAS, are they LD fluid, meaning from different independent loci, or they?
Yeah, I believe part of the pre-processing was to do initial LD pruning and then going and running your GWAS. Yeah. Because you're right. Because we also made sure that the 1,000 weren't like all within Titan, for instance, because that was because at the end, we are remapping the SNPs back to their respective gene, because then we're silencing the gene, right? So we wanted to have multiple things to consider versus just the Titan. Yeah. Mm-hmm.
So the like there's whatever 1,000.
Yes.
I know it's pretty hard to figure out how they were people. Like, are these people that were just randomly recruited and they agreed to have MRI?
Oh, good question, because we did have like completely healthy people within the sample and even doing this heart thickness. So that's actually something we should completely check, is exactly how that sampling worked, because yeah, because you would think that if they had to get an MRI and it was just part of their medical records because there was some concern in the first place, right? Yeah, good point. Yeah. We started off trying to consider HCM specifically because we know that's genetically driven, but there was only like 100 or something people. And so we're like, well, that's not going to work either. So I should say this is also after like a year of just like, what phenotype is even reasonable to work with where we'd have anything defined, right? So, and that's a part of science no one talks about. I think it's just finding the right data to start off with. Any other questions before I get into silencing? Yeah.
Identifying the genes, were you specifically looking for genes that don't have a protective effect, or was it just anything?
Anything, yeah. So even if we ended up just finding things that were actually protective, or that was also on the table. We just, because even in the gene silencing experiments, you would then see either an increase or even a decrease in size. Yeah. Yep.
You mentioned that you some inferiority.
Yes.
Wondering if there is a gold standard.
So this was not created in-house. This was an established method that had been applied to UK Biobank before to get all of these different measures from the MRI data. And then we specifically focused in on thickness after speaking with the MDs who know a lot more about what we should look for than I do. Yeah.
So,
Yeah.
So I introduced laboratories. So you have 1400 more or less independent SNP. Do you have any other covariates like sex?
So this was like essentially regressed out prior, and then you kind of like your typical, like, how you regress out the population effects using principal components, and then we had like sex and age and other demographic info as well. So this has been adjusted for age and sex. Yeah.
So the IRF will be on 6.
Yes. Yeah. Mm-hmm. Okay. All right. So this, I think, is exciting because I had no idea this was possible coming into the project. But what the researchers at Stanford did is they harvested these cells and were able to induce them to be heart cells, right? So you start off with cells from like the arm or the mouth. And then from there, they were able to silence the genes or the pairs of genes that we wanted, and then grow these heart cells over the course of a month. And so at the end, we have these cells that have been growing, and then we're actually going to look at videos of them flowing through this little pipe. And then part of my job, among others, was to identify the cell in each photo. That's quickly a snapshot is taken as it flows through this pipe. So you have to identify where it's at, and then quickly draw, like get an outline and all these other measurements of the cell. So you want to see like how round is it? What's the size? Is it really weird looking? Does it seem to be a clump with something else? So it's very, very messy. And I should say, I don't think you can see it here, but all right, the cells promised to us did not look like this. Okay. When we came to our collaborators, they were beautiful. You could see the nuclei and everything. And then they were like, here you go. And we got terabytes of this. And we're like, oh no, what do we do now? And so to actually even vet the procedure we were using to see if we were getting correct outlines of cells, this was, again, my job as a new postdoc. So I collected family labor from my aunties, my brothers, and I put them all on different sets of images and had them outlined to see how similar we all were, and then compared that with the actual procedure we were using standard image segmentation approaches. Because every image segmentation approach is based off some other experiment that people use that are baseline images and not our image, right? So even if you use some crazy deep learning model, it's going to completely fail on our data because ours doesn't fit the distribution that they're using. And so fortunately, we finally got a pipeline that seemed to match up with everyone. That also, I think, was more accurate than my annotation, at least. And so this was just yet another step of trying to vet and make sure we're not just collecting nonsense, basically, when doing this test. And so I also say this because it's important to look at all of your data as much as possible. One thing that our collaborators missed is that several of the cells would turn into clumps like this, which means they're also slower as they move through the pipeline. And so you would get this big guy stuck in your pipe, which means it gets recorded like 100 times, right? Because the photos are just being taken. And so you'd end up with these weird shapes. And it's not because, oh, my gene silencing was so good that we shrank that size down dramatically, right? Completely false. It's because we had left in these giant cells that were still getting collected through the pipeline. So even though it was a positive result, unfortunately, we had to filter those out as well. And so I say this especially because I think this seminar is friendly to it, because just trying to emphasize stable results throughout the pipeline, I think you end up sometimes hurting your results, sure, but it's for the good in the end. And you can miss out some pretty important aspects if you don't carefully look at everything. But to get to the punchline, knocking down genes, we actually did see decreases, in heart cell size in 4 out of 5 experiments. So no, we didn't get everything, but we actually did see a nice, clear difference. And what I think is really interesting, is that if you look at the upper quantiles, so we had 2 cell lines, one was from a completely healthy person and one was from someone with HCM, the heart disease I mentioned. And if you look at the most extreme quantile for each, doing the knockdown actually reduced that size substantially. So what I mean is, for the cells that matter, so for the people that would have had a very thick wall, that's where we saw the largest reduction in the size, which actually means a lot more clinically. And so talking with our collaborators, they were actually most excited about this quantile difference because it doesn't really matter if there's small reductions in like a healthy size in the first place, right? In fact, that may or may not even be good, but it's really sort of these extremes that we care about. Okay, so now this is where I'm going to go back and talk about where we might have gone wrong, or at least where, maybe a better way to put it is where we can improve, right? Because the weights that we used within the random forest algorithm kind of indicated where we got to search because it changes the distribution, right? And so if these weights are off in some way, we maybe didn't get to see everything we should have looked at. But before I get into this, does anyone have questions at this point? No? Okay, so I mentioned that relying on something like MDI might be a problem. And so let me see, when do I stop again? Oh, okay, perfect. So this is like my JSM talk, so it's exactly 25 minutes. Okay, so I mentioned MDI. This is if you ran random forest out of the box in R or Python and you looked at feature importances, this is the one they're going to show you, okay? And so it's actually known to have several issues. It's unstable in low signal problems. It's unstable with highly correlated features. It's biased towards features with high entropy, so really high variance. And it's actually not very good if there actually is additive structure. So if your underlying model is like this simple, nice linear thing, decision trees are actually not great at that. And MDI is also not good at that. And so what we're actually also going to show is that MDI falls apart if you have data dependence, if you have outlying behavior, which again could be the case given we're using these potentially noisy public data sets. And so just thinking back to the challenges in our problem, this might be an issue, right? Because we have several of these challenges in this type of data. And probably most real problems, right, will fall under this umbrella. And so this sort of incentivized, okay, can we just improve this importance aspect? Because not only would it help in the IRF setting, but in general, a lot of people just quickly run a random forest, look at the importances, and it's done, right? It's a really common, well-used predictive method, but there's clearly issues if you're applying it on real data. And so this was a joint effort with Abby Agrawal, Tiffany Tang, Yanshu Tan, and Bin Yoon. Yanshu is now in Singapore. Tiffany is starting her position at Notre Dame soon. So nice group. And so the rest of this talk, I want to highlight this connection between linear regression and decision trees. I talked about earlier, there's actually a nice way to put a lot of things into a regression setting. And as statisticians, hopefully that excites you because we love regression and we understand it. And we also know a lot of alternatives to it, right? So we can work with that and reinterpret feature importance in this framework before I think it naturally leads you to what we ended up proposing for a generalized and feature importance measure, MDI+. Okay, so bear with me here. There's going to be a bit of funny features going around, but it'll all hopefully come together, okay? So let's say we're starting off with our data matrix and we have 7 observations and 2 features, okay? And we fit a decision tree. And so what I'm going to do is create what I'm referring to as supervised engineered features or tree-based features using the combination of my data and a fitted tree. And so for the first observation, I'm going to like recode this where I look at my node 1 and I say, okay, I turned left. I'm going to make that a -1. And then I follow this down at node 2. I turned right for this observation because I have a 3 here. So I'm going to recode that as a +1. Okay. I see concerned face. Is everyone with me still? Is that okay? Okay, for the second observation, I'm going to do it again. I'm sorry if I'm boring some of you, but for node 1, I go through, I turn right. So now we're doing a +1. And then there is no node 2 after on this path, right? And so that's recoded as a 0. So that way I'm able to represent whether I continue down the path or not using -1s, 1s, and 0s. And so this wasn't the most exciting of trees, but this is what we get in the end, okay? And so this is a new representation phi of the supervised features. Now, I want you to know that this has y embedded in it because it's based off the decision tree where y is used to build this model in the first place, right? So this is not, I cannot get this with just my x's, like I had to use my y. But what's really cool about this, and this was not us, this is Jason Klusowski put out a paper that showed that if you do ordinary least squares regression on these features with your y, that's actually, you can completely recover the tree model. So these are going to get the same predictions, these 2 things. And so why does that matter? Well, you might think, okay, if I have this model here, what if I didn't do an ordinary least squared regression, right? What if I did a lasso? What if I did a ridge? What if I did any other model that may be more predictive than an OLS? Because this is just to recover predictions. So I can keep my tree structure basically, and then shove those features into some other box that you want instead of OLS. And so the question we had was, okay, given this relationship, how can I use that relationship to also reinterpret feature importances? Because MDI can then also be rewritten in a regression framework. And we actually show, well, I gave it away here, that MDI is just like using an R-squared value. Now, hopefully we all remember why R-squared values can be bad. What if I have a ton of features, I don't adjust, things gonna spike up, right? And so that's why a lot of these drawbacks with MDI happen is because it's simple regression. We know why it's gonna go bad. And so actually that regression class they made us all take is very helpful here. And so a quick reminder, if anyone hasn't seen MDI before, but the idea behind it is you're getting one measure for each feature to measure its importance. And what you're trying to do is look at how much you're getting a decrease in variance for each split on that feature. So like how much are you gaining and how much are you predicting if you split on X2, right? And so for instance, at node N2, you look at the variance here, and you subtract off everything you would be getting at following nodes. So because you split at N2, here's what your total variance is after you went down the whole path. And so then the MDI of X2 is actually the sum of all of these changes for every node that it splits on. So you're just getting just how much variation X2 specifically is contributing. But I find it easier to think about this instead in terms of our regression framework versus this change in variance. And we can actually prove that this is true. So this is not just empirical, it's mathematically sound. And so let's first rewrite our tree in terms of those -1, 1, and 0 features that I mentioned earlier, okay? And so what I'm gonna do is the stumps that split on a given feature xk, I'm just gonna move the columns over. So I'm just putting those in front. I'm not changing anything. I'm just shifting them over and sorting them so that the features for the stumps for a given feature I'm concerned about are up front. And then I'm gonna fit my linear model. And so like I mentioned, by doing this regression, you completely recover your tree, and I'm keeping the betas that come from that regression, okay? And now what we're doing is we're gonna see, okay, well, what is the partial model prediction if I only use the nodes that belonged to xk? So if I only looked at the part of the tree that xk is involved in, how does that change my prediction? And then you look at the changes between y and this partial prediction here. This becomes an R-squared value. And so what MDI is doing is it's just taking your ordinary model. Let's only take the betas corresponding to the guys that go to a specific feature and look at the overall R-squared of that instead. So it's actually very simple, just partial R-squared for MDI. And if you want to do it for a tree, you just take an average. So easy, right? Now, going back to the drawbacks that I mentioned, well, if we think of correlation, why does this fall apart? So if X1 is my true guy, it's my gene that I want to find, but it's highly correlated, or a SNP I want to find. Let's say it's highly correlated or in LD with other SNPs, right? That means the two of them are fighting over splits because the tree is only going to split so much on each one, right? And so if you're having to fight with a bunch of other correlated guys, that means you have fewer stumps here that contribute to your prediction, which means you have fewer features going into your regression, right? And so we remember if you have fewer features, right, in your regression model, your R-squared isn't going to be as high, yeah? You have fewer degrees of freedom. And so the correlated guys that got more features are naturally going to get higher ranked compared to the signal guy, just because of that correlation going on. And so it actually makes complete sense when you write it, again, in a regression framework. We knew these drawbacks implicitly or empirically from existing results, but now we actually have some like nice intuition. Okay, I won't go into additive because I think 15 minutes. Okay, so I'll get into the results. All right. Okay, so I hope that this is kind of intuitive, or at least the steps are, because I think to extend this and make it more generalizable, It's not too complicated. So before, what we were doing is we were creating these features from your tree, right? Well, there's no one that says you have to limit yourself to just those features from your tree structure itself. So for instance, I mentioned that decision trees are known to be bad at estimating additive structure. So what you can do is actually add in your original features here to help encapsulate some of the additive structure right off the bat. Or if you wanted to take functions of these features, you can do that as well. So you see there's a ton of just feature engineering, which is very CS-y. I know that they love to do that. But if someone wanted to tackle this step, you can. We did not. We left it open. But it's just like, why do we use these features? We could use transformations of them, right? You can do a deep learning model on top of a tree-based feature and then see what happens. Not saying you should or it's going to be great, but it's very flexible. Now, what I'm more interested in here is instead of doing an OLS, let's just make it a GLM, right? Let's use this because, okay, if I have a binary response, I could do, say, logistic regression. Though, as we chatted earlier, logistic regression might not matter that much in the first place. But what we actually show is, especially for things like, I don't know, for say, if you have outlying behavior, so let's say you have certain observations that were misread or something, doing a robust regression model is much better than just doing OLS, and that will improve your prediction quite a bit. So there are different options here. And you can even do a lasso, which is really interesting because that actually induces sparsity on my structure, which means you're kind of removing parts of your tree in this sort of data-driven way with a lasso on top of it. And so maybe you overgrew your tree or something, and this is removing different nodes, right? So there's a lot you can play with here, given the type of problem you have and the data that you're looking at. From there, for partial model predictions, the way we generalize is instead of keeping everything in sample, we use leave one out. So really just a way to prevent overfitting. Just, again, basic things that we all kind of know to do now to help prevent overfitting. And then finally, instead of R squared, you can at least do an adjusted R squared. You can do anything really here as a metric instead of straight up R squared corresponding to a regression model. And so on the GLM as well, for instance, there are a lot of extensions here where if you have survival data, why not you do a survival-based model here too, right? Because all you're doing is changing out your features, but your Y is going to stay the same. So you might still have structure there that you want to use. All right. So for empirical results, okay, if you guys look at the archive paper, there's like 70 pages of an appendix with simulations because we really wanted to make sure we were doing this right. I'm not going to go into all of that, but I wanted to highlight at least where we're helping correct some of these known drawbacks, and we actually saw it work empirically. Okay. And so just an overview, we are trying to compare how our method works under correlated features compared to others. And so here's our setting. We have our typical Gaussian features. We're just starting pretty small, 250 observations, 100 features here. And so we're going to have essentially 3 blocks of guides: correlated signal, correlated non-signal, and uncorrelated non-signal. Okay, so what does that mean? So correlated signal is this corner. So these are the actual 5 features that are going to be in this very basic sparse linear regression model, right? So they come from here. This is what we want to be ranked very high, meaning I hope they're in my top 1 through 5, right? That would be optimal, is if they get ranked the highest on my feature importance. Next, we get their non-signal features. So they're not part of my model. So they're not like truly causal, but they're highly correlated, and they're just as correlated with the signal guys. So ideally, hopefully they're not ranked as high, but maybe they should be ranked next, right? Because at least they're similar enough. Let's say they're in the same LD block. So maybe you at least found the right location you should go. And then from there, you can kind of expand out. So that's the hope with these guys. And then uncorrelated. So this should really be bottom of the barrel, ranked at the bottom. They have nothing to do with my signal. They're ranked last, okay? All right. And so here's what happened. So we tried multiple different existing feature importance methods. This is all random forest based. Here's us, and then we have out-of-bag versions, permutation-based versions, vanilla, and then tree shap, which is a very popular one in the machine learning community. And so what was interesting, so the dark green here is signal. Oh, and I should say, so average rank is on the y-axis. And remember, ranking means lower is better. So you want your signal features to be close to 1 through 5, right? And we cranked up the correlation. So we actually see the opposite here, where highly correlated features, my signal features get so bad that they're actually ranked worse than my non-signal. So this guy had no connection with those other features at all. But yet as correlation goes up, they gain more power. And the reason is because they get more splits naturally, while these other two groups are fighting over splits. So as you crank up that correlation, they keep fighting, which means they drop in rank. Does that make sense? So the correlation completely messes this up. And even though this guy should be an easy problem and thrown out, they actually get ranked very high. And again, this is just a default method that everyone uses, but it completely breaks apart with correlation. Now us, and I have to be fair, other methods also did very well on this setting. And part of that reasoning is that they did a lot of out of sample predictions, and the permutation test as well was pretty good. And so we helped correct for correlation, but that in part can also be done just with like sort of careful resampling so that there's less fighting among your features. But they don't always hold up, because that's not the most exciting of talks would be. We did just as well as everyone else. And all they had to do was do cross-validation, right? Okay. And so I wanted to give a takeaway for different types of settings, because we did try to cover, okay, what if we were in logistic? What if we were in this setting? And so under regression, what we used was like a default ridge regression with an R-squared metric. Under classification for our method, we tried to compare against logistic with a log loss metric. And then under data contamination, we added some outliers, and we compared against old Huber regression, very traditional approach, and a Huber loss metric. And so we also did a case study, which is in the paper. I didn't think I'd have time for it in this talk, but maybe I did, on drug response prediction as well. And so I highlight this one because you could insert other robust losses. Shout out to other work we did on different types of robust regression models. And so I'm just going to go over quickly the regression case and the data contamination case, right? Okay. We like bragging about our simulations because we claim that we're more realistic than other people. Now, one can argue, but the reason is what we did is we try to take all of our design matrices from different real data sets. Why is this important? Because a big part of what skews methods are sort of the correlation you're using, right? Especially with the way your true features might interact with one another. So we try to use basically different types of starting points with gene expression data, enhancer activity, very categorical data with like recidivism. And so we collected a lot of these publicly available data sets, induced signal using different types of analytic functions where we're targeting things that look like interactions. Because again, this is sort of going back to the original problem where we're trying to find gene interactions. And so we tried to steer away from your standard regression setting and do things that were a bit different. And so I found these pretty useful for just trying to vet whether your model can still work well, even with weird interactions present. And so the way we actually compare methods across here is using AUROC, which you might be like, oh, that's for classification, right? But what we did is we recode. So if we think of our features as we have some true signal guys and some non-signal guys, you can then calculate your AUC curves based off of like, oh, these are my true positive, these are my false positives, right? And you're trying to get a good balance between the two. And so it's kind of like classification, at least with the way we're reporting results. Okay. So what we did here, and this is for the simple regression case, is we increased the level of variance explained. So this would be a very challenging, noisy problem. This is a pretty easy problem, so you think everything would do well. And we increase the number of samples. And so in black is our method MDI+. Okay. And so as you can see here, we have this nice gap in AUROC that pretty much holds up across different noise levels. yay, everything works just by switching to a ridge term. So very simple. Instead of using OLS, we just use the ridge with some cross-validation. All right, not super exciting. What I like is the robust one. And so the reason is we added different levels of outliers of contamination. So this is 5% of my sample actually come from another distribution. So there's a clear shift. And then we wanted to see, well, how well are the trees going to do in this case, right? And so does it show up? Good. In blue and black, we have our MDI under ridge versus under Huber loss. So this is the robust one. And what's nice is you can actually see that there was a nice, clear benefit of using Huber over ridge, right? So it's not just careful tuning, it's not just smoothing that gives you this improvement. Actually picking a model that is more protective against data contamination is helpful. So if you have some hints at what type of structure your data might have, really focusing your model in on that and tuning it to that is helpful here. And we like that because, at least for me, just shoving things into another box or another model without looking at your data isn't always going to work here, right? And so you actually see that, in fact, using our approach can even hurt in some cases compared to others when there is outliers present, especially because ridge doesn't work well under outliers. And it doesn't hurt if there's no contamination. So it's kind of like you might as well try robust loss if there's any concern that there might be data contamination. All right. Any questions? Did the results make sense with the AUC curve? So the higher the better. That's just increasing your true positive rates essentially. Yeah. Okay. Yeah.
Also worried about
with the
probability, right? Yeah, how well that probability rather than just like, is that the
Oh, I see. So I guess, so we overall have like a ranking of the features. And so you do have an idea of like, are your true guys typically at least in the top amount is sort of how I see the AU ROC. It's more because like, I agree, we're not even trying to tune for a specific threshold. It's more about like, well, overall, regardless of where I set my threshold, thinking about the MDs involved, if they tell me I can only test 5 things, I'm at least pretty confident the right 5 tend to be ranked on top. Does that make sense?
Yeah.
Like, yeah. We were thinking in a very specific context, which I get reminded that's not everyone's context as I present this. So thank you. Anything else? I should say that also because we do have this reinterpretation with regression, you can get p-values too for like each beta based off the nodes that you have. And so you can even look at like the significance of a given like stump in your model, which is interesting. And we haven't actually taken a closer look there, but like you can do like tests on that too, like an F test comparing if you included the nodes going to that feature versus not, and use that to decide whether your feature is important or not. So you can do a completely like traditional test driven case. Yeah. Yeah, so here it's because like essentially your features are the only part of the random forest that's still there, and that's just to recode structure. So you're really just doing a test on new features and your Y. So you're not even having to like work with that anymore. So it's all post hoc. Yeah. I should mention we're not refitting anything. You fit your one random forest that seems to do well prediction wise. And then all of this is done ad hoc, where you go and like play around with the tree structure itself to change things. So you don't have to rerun anything. Yeah. Okay, there's no more questions, and I have too many future work things. You can tell I'm new faculty who had to come to a talk with like 20 things I want to do, right? But for those who are interested or in areas of survival, longitudinal, what have you, something I really want to get to is trying out more of those structured modeling instead of like a typical GLM. I went with easy things, but like we work on problems that have all these other structures. And so I think it'd be interesting to see how much importance change if you inserted like a model that's actually tailored towards that problem. and still sort of utilize that prediction power from a random forest. There's also like group feature importances. So I'm trying to think back to get this to work in the genetic context. And so for instance, having group sort of importances for a specific gene versus having it assigned to individual SNPs and then going back, I think would be really important and try to exploit that group structure. So you can do, for instance, a group lasso instead for your GLM. And then I won't go into everything, but thank you all for listening.
Thank you so much for these. Are there any questions in the room?
The heritability? Oh, wait, sorry, the tree stuff? Okay. This one with the ridge? Yes.
Okay, so the way we increase it from 10% to 80% is by increasing the importance of each feature versus like increasing the number of relevant features.
So the sizes are all, the data size is fixed aside from n that's changing, and then the PVE is like the level of noise added to your model. So like, I should say these are the underlying models and there's some noise level added to each. And so I changed the PVE by adjusting how much noise.
Yeah. Okay, so the number of relevant features actually remain the same. The number of features remain the same.
Yes, so they were, sorry, they changed depending on the interaction we did. So sometimes it's like 6 or, yeah.
So why the area of the curve not change substantially from the left to the right side?
With the number of samples?
No, between the figures. Yeah. You
Oh.
so various explain sounds 80%, but then the y-axis they change. So
Oh, I see what you mean. Like, why is this still around? Like,
yeah, exactly.
probably still 50. A good question.
Scale's different on the left. You look at this 0.75.
Yeah, this is a bit zoomed in on here. I should
Have
have.
one that's on the same scale between the left to the right figure.
Well, yeah, the smallest data point is probably all around 0.5 still, with very low sample size. And then some of these go up faster versus others. Well, that's a good point. You would think that they like jump up, right, with like 0.8 versus others, versus not. This was under ridge. You know, it does change depending on the X that we used. So if I remember right, this one came from enhancer activity, I believe, which had a lot of clumps of correlation in it. And so that, like, some of them jumped up much more dramatically than others. Like recidivism was pretty boring as a plot because like everybody was winning by the time you got to 0.8. Versus you saw more changes depending on the X matrix. So I think part of it is under certain correlation, even if your noise is really, even if your signal is really high, everybody's still having a hard time. And they don't quite all increase together. But I do have other ones that spiked up more. It was just an easier X value. Oh, sorry.
So I understand the like what level is
Oh, that's a good question. It also depends on the problem. Yeah. So like for the heart disease problem, if we got up to 0.6, I'd be celebrating because it's so hard and noisy, right? And then on other settings, benchmark models are like starting at 0.6, right? And onwards. So I would say that's highly data dependent. I mean, ideally, you don't like if you're at 50, you know that you're no better than random chance, right? But it's highly dependent on how low the signal is for your problem. Yeah. Uh, for this to work in general. I think it depends on the noise level, really, because like if your noise is high, then maybe you can get up to 0.7 relatively quickly, right? So I'm sorry, I don't have the best answer. I'm sure there are some better like power calculations you can do, especially with the regression setting. So you can probably do some more actual sample size comparisons. Yeah.
Thanks, I really enjoyed your talk, it was super interesting, very clear, thanks.
Thank you.
I appreciate it with diagrams. I just wondering, so if your first example of the heart disease, when you use the MGO plus compared to other sort of, the MBI or whatever, how different were the sort of results that you were getting? Were they substantial or?
Okay, so I should say that we only just finished the heart disease paper
Oh.
pre-MDI, and so this was like an ongoing
Okay.
as we were trying to get the other one working. So like I continue this now where we're trying to get it all. Basically, the scaling is the problem, because even so we have the 1400 and we can apply it directly. And it's been more of a time issue since everyone started new jobs and needs to go back and try it. But yeah, it's another thing on the list to do.
You mentioned like the slide of 4 steps of like the MDI plus.
Mhm.
Like, do you need any actions on the penalty or relationship between P and N for things to like work out? Like for the theoretical results that you're having?
So good question. So the theoretical results we have essentially are establishing like proving the equivalence of MDI to R squared, but we don't have any consistency on like the ranking of the features themselves. And so this is very needed ongoing work, but in general, random forest literature with feature importances is very weak on actual consistency theory. Like people maybe simmer down to a decision tree and try to say something there about like consistency of splits instead. And so there's a few of us that are just trying to find where to start because you're trying to prove consistency on, an X that is designed from a structure of a model. And so it's very weird, right, to work with. And so we were trying to start with like, what if you did the wall test? At least you have, you could work with like traditional inference asymptotics there and maybe work from that instead. But yeah, no, that's just trying to get to some sort of like screening guarantee of like, as long as you go up to some number, you'll get the true guys has been a headache. Yeah.
Seems like the data sets you mentioned were like mostly structured data sets. Like if you thought about like function free text or something.
Oh, interesting. Because like what the random forest on like tabular data is common and stuff. Yeah.
I don't know if you looked into like
So that we would definitely try. It's more like we had what we had access to, and we just tried a bunch of them. But yeah, that would be interesting. Anything that like if we know that a random forest performs well enough, then it's easy to just push it into this because you're really just extracting the tree structure and then working downstream. So yeah, if you have any areas that random forest worked well, I would love to throw it in and see what happens.
we're right at time.
Yay. I see 3 things in chat. I don't know. Oh, okay.
I'm gonna ask her.
Thank you both.
Okay, yeah, thank you so much. It was such a wonderful and like.
Oh, thank you.